topic by topic. We're going to have a look at homeostasis, the idea of trying to stay the same. When the conditions change, you don't. If I take you to the North Pole, where it's really cold, your body temperature is 37 degrees. If we go to the Sahara Desert, in the heat of the day, where it's sort of really hot, your body temperature is 37 degrees. And your body remains within that 37 degrees plus or minus just a little bit. And by that I mean just not few degrees, but tenths of degrees. And it stays that basically all your life. If we manage to cool you down, then we get you below something like about 34 centigrade, 34 Celsius, then you die. And if we take your body much above 40, you also die. Sometimes you raise your own body temperature so that you can kill the bacteria that might be invading your body. So homeostasis is this art of staying the same temperature. It's keeping the same amount of water in your body. If you get too much water, then you get rid of it. If you haven't got enough, then your body stops getting rid of water and tells you you're thirsty. We keep the right amount of sugar in the body. Too much sugar and your body starts to have real problems. Too little sugar and you also got big problems. So your body sugar levels stay the same. All sorts of things stay the same, and it's about this art of basically controlling the body, homeostasis. So this principle of homeostasis well, let's see what we can say about it. It is basically your response to basically, I suppose, internal and external changes to basically maintain your balance. So we've got this idea of conditions change, either can be internal or external, but your body stays the same. And we said this is true for things like temperature. It's the same for the amount of water in your body, and it's also true of the amount of glucose in your body. And basically homeostasis involves some sort of coordination of not just things but everything. Coordination and, I suppose, control. And this idea of coordination and control keeps us steady. It's not just humans that do this, but all animals and many plants do this. And to do this, basically, you need, for the control, we need receptors. We need control centers or coordination centers. And to do something about this, we need effectors. So receptors are cells in your body, internal or external to the environment, and they respond to stimuli. So we get some sort of stimuli. The receptors then may be part of the hormonal system, they may be part of the nervous system, and they send their information to this coordination center. And this coordination center, largely, most of it's in the brain, the hypothalamus, then it too sends out information to the effectors, whether they be things like muscles or glands to sort of produce hormones. And everything is done to try and maintain what we will call optimum conditions. And if we can do that, then your body is going to stay as healthy as it can. Homeostasis is important for maintaining optimal conditions for all the enzyme reactions that are going to go on, all the cell functions. It's going to control everything to keep you in tip-top condition. Now, the systems that do this, we've got basically two types. We've got a hormonal system, which is slow and tends to be more body-wide. So, for instance, if I brought a lion into the room, you have a look at this lion and you have this sudden panic. You release some hormones and they flow around in the bloodstream to all parts of your body, and they get your body ready to perhaps fight or flight, run. The lion then charges you, and you've got two choices then. You can stand your ground and you can fight it, or you can run away. And if you're with me, then remember, you've only got to run slightly faster than I can, and then basically you're safe. The other way is with the nervous system. Hormones control lots of things in the body, and they work slowly. Nerves tend to produce one particular effect, to do something, to sort of raise a hand, to lower a hand, to move a finger, to move an arm, to move a leg. They control basically all the muscles. So the nervous system is a much more specific system and a much faster system. And you can only go so fast with your system. So if I take my finger, I can move my finger so fast, but I can't make it go any faster. It just will not go any faster because we've got limits on our system. So let's take a look at how the nervous system works to start with. So we'll have a look at the structure and the function of the human nervous system. So what have we got in a nervous system? Well, obviously the most important thing here, we've got nerves, and particularly these are nerve cells. So our first question is, how big are they? And the answer is they are some of the largest cells in the body. That doesn't mean to say they're huge like this. No, it doesn't mean that at all. It means that they may be small, but they have parts that go down and reach into far bits from perhaps the end of your big toe to the base of your spine. And that's quite a large nerve. that means it's going to be, in most cases, probably greater than 1 meter in length. These cells are also extremely fine, which is why if you damage them and this gets severed, then they don't work anymore. And this is a big problem. And people say, well, why can't you sew those things together? We can barely see them under the microscope, and they are smaller than a needle. So joining them up is at the moment really just not feasible to do this. So we start off with the nerve cells, and we start off with, we've got various different receptors. Now, when we sort of ask questions about how many senses do you have, how many sort of receptors you have, we get the 5 senses. But in fact, this is way wrong. Let's look at your finger. You've got in there a temperature sensor. We've got something all about pressure. I can tell whether I'm pressing gently or whether I'm pressing hard. Hard, you saw my blood system was sort of challenged there, whereas gently it's not. Press hard. So I've got temperature, pressure. I can tell whether there's water there. I can tell whether it's wet. We can tell whether it's hot or cold. So we've got a whole variety of things, and that's not the exclusive list. But we've got all sorts of things we can basically test in our finger. And this information is transferred from the peripheral nervous system to basically the brain, the central nervous system. So we've got this idea of a central nervous system. And this is basically considered to be your spine and your brain. The peripheral nervous system well, this is basically everything that's not your spine and brain that goes round and sends information all around your body. So we've got basically 3 types of nerves. We have the sensory nerves, and what these do is they send to the central nervous system. They send all that information. We've got relay, they're called neurons, and these pass information from one cell to another. And we've got with these things like the motor neurons. These send information from the central nervous system out to the body, and these things are what we call effectors. they effect something. They make something do something. They're either going to go to a gland and say secrete something, or they're going off to a muscle where they're going to say move. Effectors then, glands or muscles. Your muscles respond basically to the arrival of those impulses and they contract. Your glands on the other hand respond by releasing, proper word is secreting, a chemical substance. So for example, you're about your salivary glands. They produce and release extra saliva when you smell the cooking, and your pancreas releases that hormone insulin when your body detects it's got too much blood sugar when you've just had a meal. So let's put this together in a plan. We have some sort of stimulus. This stimulus is picked up by a receptor. The receptor has it passed to the central nervous system, and the central nervous system will then pass this information, if necessary, back to an effector. And we need to know this off by heart. So let's have a look at something like one of these nerves to see, what does it actually look like and what does it do? For GCSE, you don't need to know too much about the detail about this, but it helps to have some idea about what's going on. So let's have a look at here. Well, I've got some sort of, let's try drawing my finger, let's give it a nail, and here I've got various different sensors, these little receptors in my finger here, and this goes along to this sensory neuron. Now sensory neurons normally got little cells around it. Now these are called Schwann cells, and they put sort of an insulation layer around this little nerve, and this is called a myelin sheath. So we don't need to know about the Schwann cells, but we just need to know that we've got this myelin sheath which does basically some insulation. This is fat, and it's stopping this electrical signal going anywhere else. It just goes down through here at a rapid pace, and somewhere off of here we've got the actual cell body where the nucleus is and all the other bits and pieces, and then this also carries on where it terminates with some sort of connector. So this is our sensory neuron. We don't need to know it in too much detail. It's got some sort of receptor down here. It's got a neuron fibre, and the impulse travels this way towards the central nervous system. Our central nervous system What does that look like? Well, really quite simple. Here is my central nervous system. We've got basically our spine. We've got an old ancient part of the brain, the medulla oblongata, and then we've got the higher parts of the brain that do the thinking, the processing. The medulla does all the things that you don't think about, the things that you might decide to sort of say, alright, I think I better beat my heart now, I better breathe in, or I'm just going to contract that piece of gut. All those sort of things that you don't think of. Yeah, you can think about your breathing, you can hold your breath for a while, but basically you don't do that most of the time. You don't think about that. That's controlled by the automatic part of the brain, the bit that you don't really think about. You try doing this now. Stop your heart. Yeah, you can't. And it's probably just as well that you can't do it either. So we've got that part. But then you've got the other part of the brain that does the thinking, that does all the sort of processing. And we have a situation where you've just sensed something, and having sensed something, you decide that you need to do something about it. So then the central nervous system makes a decision, and you decide that in response to this stimuli, say you've just seen a friend, and the response to your stimuli here, this is not a program one, this is a learnt one, is you're going to go, hello, I'm going to wave to them. So how do you do the wave? Well, to do the wave, we've got to look at a different type of nerve. And this nerve looks more like, I suppose, a holly leaf, I suppose. And it's got, coming out of it, lots of little tendrils. And these reach out to lots of other nerves, because they just don't touch one, but we have many that are touched. So this is our motor neuron cell body. And this has many ins but one out, and again it's got this sort of myelin sheath, these Schwann cells, which enable the signal to travel really quite quickly. And it comes out, and there at the end it's got lots of connectors to here, either a muscle or maybe a gland. In the case of waving to somebody, then it's probably a muscle, unlikely to sort of have a whole nervous response to that. And the direction of flow for this is only out this way. So we've got these pickups, and we've got these other bits. Now there is a required practical that comes to do this, and we've got a simple experiment that we can try and do, and we're going to have a little go at that one in a minute. And that is measuring reaction times, but more about that one in a few minutes. So we'll have a go at looking at that one, but first of all, it's a better idea to have some idea of how this works. So we've got these nerves, and if we look at these nerves, what we've got is a little fine fibre, and we've got the Schwann cells either side, and where they meet, where there's a little gap, there's a special gap, the node of Ranvier. Don't need to know this. So we get an electrical signal going in. Chemicals move in and out, and this sends an electrical pulse. And the electrical pulse moves down these cells all the way along, sending this electrical signal down the cell from the receptor to the central nervous system, around the nervous system, or from the central nervous system out to the muscles and the glands. And this is done by having this electrical signal moving along. Now, as well as having this electrical signal, what we also have is something else, and that is some sort of joint. And where the cells join, we have a special connector. Very badly drawn here, and this is called a synapse. The electrical signal comes in, and there are little vesicles in here, and when the electrical signal comes in, these move to the outside where they release their chemical. The chemical travels across this little gap, the synapse, and it binds then to the surface of another cell, and that is then depolarized and then sends off another signal. The reason for having this is so that we can have three sorts of connection. We can have one come in and go to many. We can have many come in and we go to one. And we can have one go in and one go out. So we've got these types of signals, and these are used in special nerves which are found in the central nervous system, and this is a special sort of cell with its connectors, and this is our relay neuron. And it certainly doesn't look very much like that. But we've got this idea of the coming in, the processing, the going out. And we do that with electrical signals, and we also do this with these chemical bridges, these synapses. Well, it's about time to try and do an experiment, and this is one of the few experiments I can't do by myself. And what we're going to do is we're going to do the reaction test experiment. Now, for this we're going to use a ruler and drop it. Now, you'll find if you have a good look on the web, there are some templates, whereas instead of just using a ruler, you can look up how long it actually takes for this to fall. And you can either use a ruler, and we take the ruler and we hold it up and we set it at 0, and then we're going to drop it and catch it. And you can either take the numbers, the number of centimeters, or you can put something around the ruler to work out the time. I'm not going to bother with that. We're just going to do it very simply and have a look. So let's have a look at this experiment. So what do we need for this experiment? Well, I need simply a ruler, and I put my fingers here at 0. We let go of the ruler and I catch it. Well, that spoils all the fun, because I can catch that at about 3 centimeters. Do that again, or even better, I could do it at 1.5. But I know, so we're after trying to test how this works. So what I need, unfortunately, is someone to do this for me. So I'm going to do this here, and I'm going to hold this, and what we're just going to do is I'm going to have my assistant come in, and he's going to hold that, and at a random time he's going to let go. Yeah, well, it didn't go straight that time. Give me a fighting chance. That wasn't so good, but that was 22 centimeters. We'll do one more. And I would suggest that you do a whole load of these. And that one's at 14. If you can't catch the ruler, then I would suggest that if you want to go out driving, I know for some of you that won't be possible, but if you wanted to go out driving and you can't catch the ruler, then your reactions are going to be rather slow. And I, for one, wouldn't like to risk my life going out with someone like that. If you can catch the ruler within about 4 to 5 centimeters, then your reactions are lightning fast. And most of our reactions are somewhere between some of these things. Now, having done that experiment, then it's worthwhile then having a look at trying a few different modifications to this experiment. So we've done this idea of dropping the ruler, and I'll get the right one on there. If I drop the ruler, let's just draw the ruler in. So here's my ruler. We'll start holding at about 1, so that's where we're going to put our finger here, so it's at 0. These are all in centimeters, and basically we can drop the ruler and we can measure how quick you respond to a visual stimulus, because what you've got is simply you looking here at what's going on to decide when that you're actually going to close your fingers and hold the ruler. Another good fun one of these is to take a whole load of people, and I can't do this one here, and we stand these people in a circle. Here we are, so there's our circle, there's our people, and what we do is we all hold hands. It's usually not a very popular lesson, this one, and what we then do is normally one person starts this and everyone has to close their eyes, holds hands, and this person has a stopwatch, and as they click the stopwatch, this person can feel them do that, and they squeeze the next person's hand, who squeezes the next person's hand, who squeezes the next person's hand, and this person stops when the person next to them squeezes their hand. And you can measure the time it takes for the signal to go round the circle. So we've got a ruler drop, and I've got this so-called friends in a circle type of timing system. Once we've done this, then, and I don't advocate drinking here, but what I normally do is I get some of my students to have a drink, and what we'll do is we'll give them some sort of caffeine drink, and we give them a caffeine drink, usually a couple of those, and then we get them to repeat the test. When they repeat the test, we find that they've got a significant improvement in their reaction times. So then we do another one. I take a student and I let them make a phone call. And when they make a phone call, I normally get them to ring not one of their classmates, but someone else who's going to actually have a conversation with them. And this person actually has to have a conversation. And whilst they're having this conversation, someone does the ruler drop, and we drop the ruler. And invariably, they can't catch it at all. So, on some stimulant drugs, and caffeine's an acceptable stimulant that we can use that with exciting things such as coffee, tea. There are some special fizzy sort of caffeine drinks that you can buy, and we can try this, and we see an improvement in reaction times. If I get the student to take a phone call, or worse, I can get them to try and relax, then we find here we've basically got very slow reaction times. So we can measure the difference in reaction times simply by doing this. Now, it isn't a case of doing this once, but you've got to do these tests at least a minimum of 10 times, and I would normally reckon on doing more than. And what we can do is we can then draw out a little chart of the students, and we can measure their reaction times. And this is without drugs or anything, and we find that most students have a reasonable reaction times, and there are some students who you just wouldn't want to go out with. They're basically dead, I suppose, really. They can't manage to react in time, and this is a bit of a problem, especially if you've got a situation where you're in your little car and you're speeding along, and at that moment a child steps into the road in front of you. And what we have in our highway code is simply this idea of a thinking distance and a braking distance. The thinking is based on this idea of how long it's going to take you to realise that a child has stepped into the road and you are going to have to press the brake. And that might be 1 second for an average person. That does mean there are going to be some people who are going to be up here. Some of the best people I would suggest around here are probably the racing drivers. They're good, they're trained to work at this sort of speed. And then we get some other people who are much, much slower. And these, you probably know them in your class. These are the people who are asleep. Yeah, they're with you, they're there in body, but their mind is certainly elsewhere. And this thinking distance is an optimum here. This is the average. If you're a racing driver, this is going to be less. If you're asleep, then this could be an enormously large number. You then press the brake, and how hard you press the brake and how good the car is this braking distance. And this is fairly standard. Unfortunately, this one can be either short or very long. And it's about this measuring this reaction time that we get some idea of the experiments. So these experiments we can do all sorts of things with. We can try someone listening to music. We can get them to be in conversation with somebody, and we find their reactions go down a lot. It shows that if you're in a car and you're driving, then being on the mobile phone to somebody is not a good idea. But also chatting away to someone in the car might also not be a good idea. Because if you're not paying your full attention, then when a child steps in the road in front of you, that 1 second might be a little bit more. And if it might be a little bit more, then they may end up a little bit dead. Reaction times are important, and this ruler drop is an artificial way of trying to do it, but it gives some sort of measure of how fast your reactions are. And these reaction testers do give some sort of indication. We're all different. Some people's reactions are quite quick naturally, and other people's are a little bit slower. And then you worry about them if they're driving you around. If they're on some drugs, caffeine, I mean legal drugs here, then they might be a little bit better. They might, however, be on some sort of drugs that might slow them down. They might have had some nice cocoa and be nice and cosy when they're driving, and they've got the heat up, and it's nice and pleasant. They're listening to some music, and they're driving along really quite relaxed, and then something happens and they have to change. Or you've got the person that's perhaps hypertensive, holding onto the wheel, looking for everything going on. Who knows? That's what the driving test is all about. So, reaction times, something we can probably have a go at. So, there we are. That's a little introduction to homeostasis. Next time we're going to have a look at the reflex arc and what that does. So, until then, I'll see you next time on GCSE Biology Topic by Topic. So stay safe. Bye bye.